So this has to do with cancer. So if you know anything about cancer, cancer forms by a buildup of cells within your body. But the important part is the buildup of cells is not what's going to kill the patient or cause any medical problems. What's actually going to cause problems is when those cells are able to leave the original tumor site and migrate to distant sites. And the matrix surrounding the tumor, all the proteins surrounding the tumor have actually been shown to influence how cells are able to do that. And you can see it in the diagram up there. You have an increasing in the protein stiffness and also an increasing in the amount of protein and the alignment of the protein around that tumor that helps facilitate cellular migration away from the tumor. But the problem is the current technologies that are available to study this phenomenon are basically endpoint assays. So you get a reading at the beginning, you get a reading at the end, and you're not sure what's happening in the middle or over what time course that they're happening without doing repeated studies. So what we're trying to develop is we're trying to develop a very small force sensor that we can place within that tissue so that you can measure these changes as they are happening. And we're trying to use DNA origami to do that. So DNA origami is a technique that basically takes advantage of how DNA binds. So DNA binds very specifically, and what they can do is you can use that binding and a little bit of thermodynamics and control the shapes that the DNA forms. So people have made block O's, people have made smiley faces, you can make triangles, you can kind of make whatever you want out of this. What we chose to make here is you can see this structure. It's 2 rods and then a linker. Each rod for reference are about 50 microns, so they're very small. And then this linker actually has a fretting signal, and fretting is basically a technique used to measure distance. So what we can do is we can be able to tell if these barrels are very close together or very far apart. And the idea is, what we're currently at in the project, is to characterize when we see these signals are very close together and when we see these signals are very far apart, and basically create a library so that we can use this device in actual tissue. So you can see an example of the curves we're trying to create here with different weight percents of a polymer, and then with different concentrations of a polymer you get very different fret efficiencies, which is basically a metric we use to measure the distance. And that's all I have.